Okay, so I thought I would just record this talk of mine from last year's Australasian Society of Continental Philosophy conference from 2024. This is a way of getting into uploading some of my own content independently to YouTube. Now, I thought I might rewrite this talk or add some content to it or whatever, but I decided to just go ahead and record this. And this can perhaps then be seen as a prelude to some videos I'll hopefully do next year, which will be both a much deeper exploration of some of the concepts surrounding the notion of machinism, which are coming from Deleuze and Guattari, and how they, in my opinion, can be used as a conceptual lens for thinking through certain issues in biology and perhaps other sciences as well. And there'll be a contrast there with um, things like Georges Canguilhem's critique of mechanism, and of course, a lot of Gilbert Simondon's work, things that I have explored a lot in other talks, but are not in this talk. At the same time, this can be a little bit of a prelude to me revamping a series of lectures on the human relationship with drugs that I taught for a number of years at the University of Melbourne here in Australia. And those talks were predicated on taking a view from chemical ecology on pharmacology and on, again, the human relationship with drugs. So hopefully that stuff I'll do next year. But for now, this is Machinic Pharmakoecology. And that eccentric, to say the least, looking individual there is, of course, Félix Guattari. Okay. So there'll be a number of quotes throughout and some unpacking of those. And again, I haven't given this talk for over a year, so we'll see how we go. So first quote is from Anti-Oedipus from, of course, Gilles Deleuze and Félix Guattari. Quote, "It is not a matter of biologizing human history, nor of anthropologizing natural history. It is a matter of showing the common participation of the social machines and the organic machines in the desiring machines. Molecular biology is itself schizophrenic, as is microphysics." So this is just a nice orienting quote for the fact that I'm going to be talking about chemical ecology and thus molecular biology, using some of these concepts borrowed from Deleuze and Guattari, and of course, particularly the machinic. And also, it's just a note to begin with that this is not a matter of being reductive. It's not a matter of, assimilating the philosophy of Deleuze and Guattari to some sort of biologism or scientism or whatever. It's another lens. Like, it's not a matter, as they say, of biologizing human history or of anthropologizing natural history, but it is a way of finding a kind of common and, I would say, operational language for thinking about these different domains in which machines and machinic processes appear. And in this talk, of course, again, that's going to be in the world of chemical ecology. So what exactly is chemical ecology? Ecology is shaped, coordinated, managed at all levels using molecular means. Not exclusively, obviously, but molecular means are very important. So from pheromones to toxins, organisms use what I call exochemicals, and exo means on the outside. Exochemicals to influence and manipulate one another's behavior and indeed even one another's morphology. So an exochemical is a chemical produced by one organism that really has its functional role outside the body of that organism that produces it, like typically in the body of another organism, a target organism. Everything from bacteria inducing choanoflagellates to form multicellular colonies, as you can see in the picture here from the King Lab, and myriad marine invertebrates to undergo metamorphosis. There's an endless list of really fascinating examples that I could bring up here. And again, perhaps I'll do some more detailed talks next year where I go through a lot of examples. The format for this talk when it was originally given was that I only had 20 minutes. So I had to, be very light on examples and everything from that sort of thing, which is really common in nature. Like it's really important to emphasize how ubiquitous it is, really, especially in marine environments, but certainly very far from exclusively, that organisms require often metabolites or effector molecules, or they can be called lots of different things that they get from other organisms in order to complete their own life cycle. So a lot of organisms that undergo metamorphosis, again, it's very, very common for marine invertebrates that they won't undergo a transition, say, from a planktonic larval stage to a sessile and benthic, so stationary and living on the bottom, adult phase until they encounter a surface with, bacterial microfilms on it that have the right concentration of certain effector molecules that those organisms need in order to undergo their own metamorphosis, in order to undergo that transition to the next life stage. Like they cannot do it without those effector molecules that they get from symbiotic microbes. And this also is the case with a lot of terrestrial insects undergoing metamorphosis, et cetera, et cetera. This is just really, really very common that lineages kind of fuse. We're not talking necessarily about so-called endosymbiosis, such as we see in the origin of the eukaryotic cell, but we're seeing various degrees of chemical symbiosis, which are everything from sort of antagonistic relationships, which we'll talk about a bit in this talk as well, from like a venomous organism to a target organism, or a parasite. Parasites are often venomous, to a target organism. Everything from an antagonistic relationship through to a mutualist relationship where two organisms live alongside one another, and both of them benefit. And of course, in the middle there are also commensal relationships where one organism is benefiting, say an organism that can get an effector molecule from a bacteria, but it's sometimes uncertain whether those microbes are actually benefiting from living in the so-called host organism, or really that's just, like any other environment they might live in. So lots of questions to explore if I end up doing more chemical ecology videos, which I no doubt will do. But everything from that, which is sort of classic chemical ecology, to the usage of molecular tools, drugs, by humans. That's part of chemical ecology as far as I'm concerned. I refer to it as the chemical ecology of the human. And remember, again, this is sort of a prelude to me revamping and releasing my lectures on the human relationship with drugs, which very much take this kind of perspective. The tool use is literally economics, right? That's why I had the reference to managing of ecologies up there. Ecology, oikos, logos, right? The study of, or you could say the order of the home, oikos, the logos of the oikos. Economics, nomos, oikos, oikos nomos, the management of the home. And one of the major ways that organisms manage their homes is using tools, and molecular tools, again, are amongst those. And human pharmacology and drug use is very much of a piece, as we'll see with the kinds of things that a lot of other organisms do. We do it in, fancy pants human ways, as always, but it's not categorically distinctive from what a lot of other organisms do, in my opinion. So you can think about this tool use, including molecular tool use, as a kind of, as a manifestation of the notion of an extended phenotype, which Richard Dawkins introduced in 1982. In a very good book, by the way. We can bag out Dawkins, and I will often do, for his neo-Darwinism or his neo-Darwinian fundamentalism, but he had a lot of great ideas too, and he's a really good writer. So for Guattari, this is also a way of extending the limits of the machine to the functional ensemble which associates it with man, and we'll get into that a little bit more with man, but of course I'm saying with other organisms as well. Humans are far from the only tool-using organism. And this is a very Simondonian line, by the way, here, that that Guattari is giving us, and that's going to be in the subtext of a lot of this talk. I'm not going to talk about Simondon in any detail. He's very much my guy in recent years, but I kept him out of this talk. Okay, so the chemical ecology of the human, which I've mentioned, and this painting here is The Joy of Life by Max Ernst, one of my favorite artists, surrealist artist. Uh, Chemical ecology, this is just a kind of generic definition. Chemical ecology is the study of chemically mediated interactions between living organisms and the effects of those interactions on the demography, behavior, and ultimately evolution of the organisms evolved. Chemically mediated interactions between living organisms. Organisms using chemicals to mediate their interactions with other organisms. That is chemical ecology. Drugs, of course, influence interactions between humans and also the organisms that make bioactive molecules. So the kinds of co-evolutionary and ecological relationships that we have with drug-producing plants and fungus, for example, could be something we can talk about in this context. And again, that is something that I go into in those lectures that I've mentioned a few times. Of course, yeah, drugs influence interactions between humans. So the human sort of, social ecology, drugs play a huge, and often sort of underappreciated role, I think, in mediating those relationships between humans. Because it's certainly not just about, illicit drugs or recreational drugs or prescription drugs. It can be about, can you get up and get out of bed and go to work in the morning without your cup of coffee, and then if everybody needs to do that, or most people need to do that, if you're in a sort of caffeine culture, what other effects on interactions throughout the day does that sort of pharmacology, that sort of chemical ecology influence? Okay, I've said this. Drugs are a core component of human chemical ecology. Okay, obvious. We can always ask this question, which I like to ask. I used to like to ask my sort of pharmacology students, is there a human without drugs? Drugs are kind of like music or religion. Pretty much every known human culture has had a relationship with some bioactive molecules. We often call this study of the anthropological study of the relationship of sort of non-Western cultures with bioactive molecules, ethnopharmacology, because we're always marking out every class other than the Western as, the marked classes. Anyway, that's not what I'm going to get into here either, but it's just the norm. It's just a standard pattern of human behavior. It's almost like what we would call in evolutionary biology or evolutionary ethology, a synapomorphy, a behavioral synapomorphy of different human cultures that they have relationships with drugs. And for much of human history, those have been drugs that have been found in the immediate environment of the cultures that use them. So it's very much an ecological thing. Okay, here's some more Guattari from Machinic Heterogenesis, chapter 2 of his book Chaosmosis, which I lean on a lot here. Quote, common usage suggests that we speak of the machine as a subset of technology. We should, however, consider the problematic of technology as dependent upon machines and not the inverse. The machine would become the prerequisite for technology rather than its expression. This is really interesting, and this could be unpacked in great length, And we could also talk about the difference between the sort of ensemble of technical objects, so the ensemble of all the tools and machines and whatever, and technology, as again Simondon makes that distinction. Technology is the logos of techne for Simondon, right? It's the study of techne, of technical objects, and of course for him their genesis, so that's technicity. Here, really, the way I'm going to use this quote is to leap into a discussion of the fact that like machinism is a kind of generic feature of processes of production for Deleuze and here Guattari. So it's not just limited to technology, which may here be referring to the ensemble of technical objects, but they are specific manifestations of this machinism or machinic activity. So We're going to talk about toxins as technical objects here. Um, and, going on from that Guattari quote, suggests that the fact that nature is itself machinic is a prerequisite for human techne. So nature is already in the pre-human or the non-human domain, already machinic. And again, like this could have turned into a really, really elaborate or detailed discussion of the notion of machinism for Deleuze and Guattari. I don't think that's where I'm going to go right now, but maybe there's a little bit later in the talk where I'll get into that a bit more, but also other talks. So development of technical objects is indeed often reliant on accumulated natural work. So the literal historical development of technical objects is often downstream of natural work. And indeed, we often have taken, and this is sort of a Kungian point, but a more generic point, have taken certain quote unquote natural objects and natural operations, things that say other organisms are doing, or even things that the human organism is doing, and we have modeled the development of technical objects and mechanisms on those. Techne is also a form of mediation. Uh, so niche construction, for example, management of the niche, which I've already mentioned, and these are very much, pre and non-human um, processes as well. Now, toxin, like drug, and indeed pharmacon, classically, the pharmacon, which has, of course, both positive and negative effects, it's potentially both poison and cure, it's an ambiguous term. So toxin and drug are very ambiguous terms. In biology, though, toxins are molecules that are used in antagonistic relationships between organisms, used to mediate antagonistic relationships between organisms. So they tend to be used for defense, uh, feeding, or competitor deterrence. So poisons are often used defensively to prevent one organism, the poisonous organism, from being eaten by a predator who's hopefully not resistant to the toxin, as we'll get into in a second. Often used in feeding, right? So both a lot of parasites, including things like hematophagous insects like mosquitoes, um, or or hematophagous worms like leeches, many other parasites, arachnids like ticks, blah, 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 use toxins to facilitate feeding. Um, Everything from, anticoagulants to anesthetics in those toxins. And of course, parasites, many, many different parasites do all kinds of crazy stuff, right, with toxins, which I'm not going to get into here, but so many cool examples that we could get into. But of course, venomous organisms as well, like snakes, spiders, scorpions, others, they use their venom, which is a specialized type of poison, as we'll see, to subdue, typically to subdue target organisms in order to make them easier to feed upon. Swallow whole in the case of a snake, and competitor deterrence, like venom and other noxious chemicals are often used as forms of competitor deterrence. And like a cool example, for example, of a venom used in this way would be the venom of the male platypus. So not everybody knows that the male platypus, not the female, has a spur on his hind legs, spurs on each hind leg, connected to a venom gland. And in combat, territorial conflict, male platypuses will in fact sting one another, sort of testing one another's pain tolerance, I guess. It's a very painful venom that they have. So basically, there's an ambiguity here, which is not just, linguistic or semantic, surrounding things like toxin, for example, and also drug, but there is definitely a semantic ambiguity. But there's also a context dependence of toxicity and bioactivity. For example, what's poisonous to one organism is not necessarily poisonous to another, right? For something to be a toxin, it'll have to exert its toxic effect in a very specific context. And this becomes really fascinating to think about in these phenomena of toxin resistance and toxin sequestration. So I've got some very colorful looking animals on this slide, and they are colorful for a specific functional reason. These are aposematic colors. Aposematic means warning. So clockwise from the top left, we've got the wanderer or monarch butterfly. Then we've got the tiger keelback. We've got this pajama nudibranch, a Chromodorus nudibranch, and we've got this pumilio, this strawberry poison dart frog. And all of these animals are poisonous, right? This snake, by the way, is an interesting case because it is both venomous and poisonous. Uh, it's poisonous in two different ways, we could say. But all of these animals get the toxins that make them poisonous, so make them poisonous to feed upon. And we will, I think I've got a slide where I disambiguate poison from the more specialized, more restricted class, which is venom. If not, I will talk about that. The toxins that make these animals poisonous to eat were not made by them. They're not encoded by their own genomes. They are all sequestered from environmental sources. So the milkweed in the case of the butterfly, toads in the case of this tiger keelback, this snake, it's really cool, right? It eats toads, which it subdues with its own venom through biting them. And this is a snake that is potentially capable of giving a lethal bite to a human, although it's a non-front-fanged snake. Again, details, which I love, but I won't get into. The venom gland, by the way, is a post-orbital dental gland. It's on the top jaw, behind the eye, basically, or under the top lip. And it is connected in these guys to a large, an enlarged rear tooth at fang, right? A rear fang, a fang at the back of the mouth, but it's an ungrooved fang. Snake gland and dentition morphology is just endlessly fascinating and one of the areas I've done tons of work on. But it's got that. So it's got that venom gland, but it's also got these snoocial glands, which are in the neck of the snake. And they're actually right underneath these black and orange bands here, the aposematic coloration that says, I'm poisonous, don't eat me. And when this snake eats a toad, it actually sequesters the toxins from the toad that make the toad so poisonous. Of course, the snake is resistant to those toad toxins. You have to be if you're going to eat a toad, or you're going to make a habit of eating toads. It then sequesters them. It stores them in specialized glands in its neck here, making it poisonous in that sense as well. The Chromodorus nudibranch eats sponges. It's actually here pictured on a sponge. This one. It eats sponges and sequesters toxins from the sponge, and these poison dart frogs eat ants and get their toxins from ants, right? So this is one organism appropriating another organism's bioactive molecules for its own use. Maybe you can see where this is going, right? How does tool use begin in nature? All right, so this slide is titled Toxins as Technical Objects. And humans do something very similar to what these organisms do. We call it pharmacology. Nature, right? Other organisms were for the longest time, the primary source of the bioactive molecules that we used as drugs. And as a matter of fact, all of our pharmacological knowledge is downstream of those, they would be called lead compounds these days, but of those molecules that we found in nature's pharmacopoeia. So this is what I say when I say, what I mean when I say development of technical objects, this is an example, is often reliant on accumulated natural work. There is a natural pharmacopoeia. There's been an incredible amount of evolution of bioactive molecules in nature, and we have taken advantage of that. And we would know nothing about bioactive molecules had we not proceeded to learn about them in an ecological manner. Okay, so toxins as tools, right? This is where we're going with this. Tool use is really common in nature. There is this old sort of typically anthropocentric thesis of, humans as the tool-using species. Of course, we all know, hopefully these days, that is, yet another fantasy about our specialness or human uniqueness. And again, I'm not saying humans aren't unique, but capuchin monkeys are pretty unique too. Yeah, humans are cool. Humans are special. Like, I'm a human. I dig humans. But, this capuchin monkey is using a rock, and you can't see it in this picture, but it's using that rock to crack nuts, which are sitting on another rock. So you've got a kind of hammer and anvil type situation here. There's a corvid there, a crow of some type, using a stick to probe for insects underneath the bark of a tree. There are myriad examples of tool use of this kind in nature. Tool use is a form of exaptation, if one is familiar with that concept introduced by Stephen Jay Gould and Elizabeth Verber. Um, Exaptation is basically when something that evolved for one purpose or no purpose at all is co-opted for a different purpose, right? So you find a rock in your environment. That rock has an evolutionary history, right? Geology is the study of the evolutionary, history of rocks. Um, it evolved, but it didn't evolve for the purpose of cracking nuts, right? It would be somewhat absurd to imagine that it did, but it has certain properties that give it certain propensities for being good at for the task of for the function of cracking nuts. So the monkey, the capuchin in this case, finds a nice flat rock, takes it over to a big rounder rock perhaps, and together it's got this hammer and anvil. Neither of those rocks evolved for the purpose to which the monkey puts them. That is a form of exaptation. Toxin sequestration, this is obviously my point here, is a form of tool use. So you find a rock, you find a stick, you repurpose it for some other function, or you find a molecule in your environment and it has some bioactivity that you find useful and you take it for some completely other purpose. Now, maybe both purposes, maybe it was a defensive toxin in the life history, well, in the evolutionary history of the organism that made it. It's not, in fact, a tool for them, it's a trait. Um, and then maybe it's also a poison in the life. Its ecological role, its functional role is a poison for the organism that sequesters it. So you think, oh, is that even exaptation? That seems like the same functional role. But remember, that toxin certainly did not evolve to be a defensive tool for the snake that took it from the toad. That is the last thing that toad wants. That toad wants to be well defended against that snake. This is like adding insult to injury, right? Firstly, the snake is resistant to the toxin, then it takes it and uses it for its own purposes. I mean, that's just cheeky, isn't it? All right, so this is very much a form of exaptation. And obviously, pharmacology is the same kind of tool use, right? From tool to technical object to proto-machine, I said there. Don't actually know what I was talking about there, so we'll move on. But it is a prelude to this next slide of quotes, further quotes from Guattari. So, quote, for each type of machine, we will pose a question, not about its vital autonomy, it's not an animal, but about its singular power of enunciation, what I call its particular enunciative consistency. Yeah, this is cool, right? I mean, there's a lot going on just in this little quote. Um, there's a sort of a reference, because you'll get into a bit of a critique later of autopoiesis, and there's a reference to this notion, a critical reference to this notion of autonomy here. Machines, he doesn't want to confuse machines and animals, right? But I think, really the machinic is this general class of operations for Guattari, and organisms certainly include a lot of machinic operations in themselves. But machines in general, let's just say here, are not animals. This is not an autonomy thesis like autopoiesis claims to be, right? But erroneously, there's a suggestion here, and we'll get to the critique of autopoiesis in a minute. But it's singular power of enunciation. So it's really just about like what it can do, what it can say bring forth, but its powers of expression, what in fact it can do. This is sort of how we will taxonomize machines here in terms of their operations, their expressions, their functions, right? We're going to talk about function classes in a moment. So this is a prelude to that. Abstract machines are montages capable of relating all the heterogeneous levels. The abstract machine is transversal to them and will or will not give these levels an existence, an efficiency, a power of ontological auto-affirmation. The different components are swept up and reshaped by a sort of dynamism. Such a functional ensemble will hereafter be described as a machinic assemblage. So basically, the abstract machine here is capable of sort of concretizing, to sort of subtweet Simondon again, and a machinic assemblage. It relates. It's what might be thought of as a virtual governor in some contexts, but it relates these different machinic elements, which are not intrinsically related to one another, but it relates them according to a schema of functioning. So they come together. They're swept up and reshaped by a sort of dynamism. Indeed, that's a telos. They become orchestrated and oriented towards some particular function. And this functional ensemble is known as the machinic assemblage. And as we'll see, there is no function, or there's no speaking about what a machine can or can't do outside the context of a machinic assemblage, or a machinic element, I should say, outside the context of a machinic assemblage. Machinic elements do nothing on their own. This is a fully relational, it's really a relational ontology that we are being given here. So it's an assemblage of possible fields without any notion of generic or species relation. So if I remember rightly, the next slide is going to be about function classes. And this without any notion of generic or species relation is important there. Now, I have to say, I sometimes think that, Deleuze and Guattari underestimate, they're very keen to critique a sort of Aristotelian or Porphyrian hierarchical classification and grouping of organisms in the genus and species system, but that's still very useful. Like phylogeny is really, really useful if you want to understand how organisms got to be the way they are and why they have the particular powers of enunciation that they have. But you can also group organisms according to function class, according to their membership. You can group traits according to their membership in certain function classes, and that's also really useful. So in A Thousand Plateaus, they will talk about how the draft horse and the oxen are more closely related to one another than the draft horse is to the racehorse or whatever. And indeed, according to a categorization by function class, that would be true. But of course, the phylogenetic classification is still really interesting. It's where the two things intersect. So while you're able to realize, using a phylogenetic perspective, for example, you're able to see that venom as a trait, which is the membership of that trait in the function class venom, which we're going to talk about in just a moment, has evolved over 100 times independently just amongst animals. So I always joke about this because, philosophers always seem to think like carcinization is interesting or something. Oh, everything's becoming a crab. It's like, yeah, that's happened like 4 or 5 times independently in some relatively closely related groups of arthropods, of crustaceans, even of decapod arthropods. Venom has evolved over 100 times independently, right? So if you want interesting examples of convergent evolution, there are many. Anyway, I digress. So what is venom? This was my sort of like introduction for philosophers to venom. And I don't know if he's going to watch this, but my dear friend Jason Tuckwell has a bit of a latent arachnophobia, and I didn't give him a trigger warning about these slides and the inclusion of this photo of a funnel web that I took near my home this was in the rainforest near Mount Glorious, near where I used to live in Mount Nebo. Missed that place. This is a funnel web spider, right? And it's a male funnel web spider, and the males of the funnel webs are the highly venomous ones. And if you can see the tip of the chelicera, the tip of the fang here, there is a big drop of beautifully clear venom dribbling from that. Funnel web spiders are like one of the easiest animals, venomous animals to milk, right? To extract venom from, because all you need to do is like blow on them. And they get all hoity-toity, they get all, hot under the collar. They rear up like this, and they can be quite bright red sometimes underneath. Again, that's a kind of aposematic coloration. And they start dribbling venom. So all you have to do is then pipette that venom off the tip of the fangs. I wasn't out collecting venom on this particular occasion. I just flipped a log, and there was this beautiful funnel web, and it duly gave me a beautiful defensive display. So I just snapped this quick shot. But what is venom? It's a functional trait. You can't speak of venom unless you're talking about function, right? What's it for? Is the question we're asking. It is for feeding, defense, and competitor deterrence generally speaking. We can subdivide those classes, like again, I talked about, the use of venom in different kinds of feeding, like hematophagous feeding, versus what something like a funnel web or a snake would do, which is subdue the prey animal that it was feeding upon and kill it, even though venom is not really for killing per se. Like when organisms are killed by venom, that is a kind of unintentional by or unselected byproduct of an effective way of subduing that prey organism. Snake doesn't care if the mouse is alive when it's swallowing it, as long as that mouse is easy to swallow, right? It just so happens that making them easy to swallow is often a way of killing them. It's delivered by a venom delivery system. So this is a key thing here as well. So fangs, stingers, etc. So look, venom is that specialized subset of poisons which is actively injected or introduced by the infliction of a wound into the target organism. Whereas like poisons more generally in nature are often passively absorbed or they're poisonous by ingestion. So people say things like, if it bites you and you die, it's venom. If you bite it and you die, it's poison. All right. Yeah. I've forgotten the exact phrasing of that. It's an exochemical adaptation, right? So it's a cocktail, in fact. So venom is not generally speaking, is not a simple mixture. It's not like a single molecule, right? The constituent molecules are called toxins. Um, Venom often contains many, 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 many types of toxins in a single secretion. It is, in fact, a secretion. Venom is a secretion. But it's a cocktail, so it's often a mixture of chemicals called toxins, but also other things as well, designed to function outside the body of the producing organism, in the body of another organism, target organism. It contains molecules that disrupt target organism's physiology. Those are toxins. Again, disrupting the target organism's physiology in order to facilitate feeding, defense, competitor deterrence, right? A venom system now is a complex adaptive system. So the venom system includes the fangs, includes the glands, and you could say it also includes the behavior associated with inflicting a bite and injecting venom and prey handling behavior and all this kind of stuff. It's a complex adaptive system. It has non-linear properties, and all components may exert reciprocal effects or selection pressures on one another. So not going to get into this either, but the type of venom that is effective is, for example, influenced by the type of delivery mechanism that an organism has. So if you have really big fangs or really little fangs, that might influence which types of toxins are going to be more effective, because it has an effect on which tissues they end up getting injected into in the target organism. And it gets really rich and really complicated trying to think about these sorts of things. Like, you could think about two different kinds of snakes that feed on the same type of lizard, but one of them feeds on that type of lizard at night when the lizard is asleep. So the lizard is inactive, it's cold, its heart rate is very depressed, its peripheral circulation is very, very decreased, and the snake encounters it in that state and injects venom into it. And another snake that feeds on the same type of lizard, but tends to hunt it during the day when that lizard has been sitting in the sun and is running around and its little heart is going crazy and it's got a lot of peripheral blood perfusion and blah, 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 right? And it may turn out, indeed it does turn out, that two different toxin mixtures are likely to be exhibited by those two different snakes, because same target organism but different target physiology. So these sorts of context dependencies get very, very rich when if you're really trying to answer evolutionary questions, functional questions, like why does the elephant, have a long trunk? So some sort of Rudyard Kipling just so story type thing. The complexities are really manifold, and they just ramify. Lots to talk about there, but that's not what this talk is about. Abstract machines and function classes, right? This is a baby puff adder. Isn't it cute? Look at those fangs. Look at those fangs. Yeah, amazing, right? Venom is a function class. I've kind of already introduced this notion. So it's homoplastic. Homoplasy is convergence in form and function. It's not necessarily homologous, which means it comes from like direct shared descent. So all the venom systems, for example, of viperid snakes, like this baby puff adder, that the venom system and its particular arrangement is a synapomorphy of the viperid family. So all those venom systems are homologous. But the venom systems of vipers and that funnel web that we just saw are not homologous. They're not, phylogenetically related at all, but they are homoplastic. They are convergent in form and function. Hence, they are members of the same function class, which is venom. Like I said, there are over 100 independent evolutions of venom. Take that, carcinization. There are actually way, way, way more than this. So I said here that there are greater than 100,000 species. I mean, there are more than 100,000 species of hymenopteran insect. All right, and the vast majority of hymenopterans, that's bees, ants, and wasps, are venomous. And all of the apocritin, which is most uh, hymenopteran insects, are descended from a common venomous ancestor. I'm just bulking because I was trying to think of another term there, but it doesn't matter. Um, yeah, there are probably millions of venomous organisms, and especially now we are exploring more permissive definitions of venom. Or really, it's the same definition, but we're expanding our application of it. So if you think of a microbe like a bacterium that uses a particular molecule to punch a hole in the cell wall of a target microbe and then kind of spews a bunch of toxins into the internal cellular space of that target organism through that hole that it's made. I mean, that sounds kind of a great deal like venom, right? That is venom, I think, by any reasonable definition. So we end up with just a vast, vast number of venomous organisms. Like you might think of venom as sort of special or something, like it's like, whoa, snakes, but no, it's really, really common. Um, Venom systems are concrete machinic assemblages, which is sort of what I was getting at with the complex adaptive system thing on the previous um, slide. But Venom function class is an abstract machine. Compare this also with a diagrammatic schema, which someone like Anne Sauvignard would say that the diagram as it appears in Deleuze and Guattari's philosophy is the same as the notion of an abstract machine. You can think of this in terms of a map of relations. Um, I've just referencing Peirce, of course, one of the major originators of the notion of the diagram in this sense, and also Poincaré, who has this great quote about how mathematicians aren't interested in objects, they're only interested in relations between objects, and you can substitute relations arbitrarily, substitute objects arbitrarily, as long as the relations remain fixed. Mathematicians don't care because they're not interested in matter, blah, blah, blah. That's like a diagram, right? This is Deleuze on Foucault. The diagram is a non-unifying immanent cause that is coextensive with the whole social field, right? So I'm saying there's something strongly akin, building from the Guattari quotes a few slides ago, between the diagrammatic schema, the abstract machine, and the notion of a function class, right? A way of grouping organisms according to or again grouping traits, I should say, according to their functions. And so that means that the function, which the complex assemblage, the machinic assemblage, then the concrete machinic assemblage of the venom system, is a way of relating all of the different elements of that system and orienting them towards a common purpose. That, of course, has big effects, for example, on selection. So this can be made very concrete when trying to think through these sorts of evolutionary questions that we are sort of talking about to some degree here as well. All right, here's a quote from A Thousand Plateaus. Quote, a technical element remains abstract, entirely undetermined, as long as one does not relate it to an assemblage it presupposes. It is the machine that is primary in relation to the technical element, not the technical machine, itself a collection of elements, but the social or collective machine, the machinic assemblage that determines what is a technical element at any given moment, what is its usage, extension, comprehension, etc. Okay, so in the molecular context, in the toxin context, again, what a thing does I mean, really in the biological context, in the ecological context, this is all relational, right? What a thing does is relational. What a thing is for is relational. It's context dependent. If you abstract a toxin from an organism and abstract it from its use in the feeding ecology of that organism, which is contingent upon, encounters with other organisms, when those organisms are in particular physiological states and at particular temperatures and all sorts of things, and are they a susceptible organism or a resistant organism, all these different things that come into play, all the complexities of this ecological assemblage that end up shaping, or if you want, determining what that particular toxin molecule, which is a machinic element here, is capable of doing. If you take it out of that context and you stick it in a petri dish in a lab, it can do other things, perhaps, right? You could do enzymology with it if it's an enzyme. You could do all kinds of different assays with it and determine its activity in those contexts. This is what we do a lot of in pharmacology, but that won't really necessarily tell you about its function, right? Just because it has an activity in this context in the lab, in some assay in a lab, doesn't mean you can read its function in the life of the organism that makes it off that activity. And also, of course, in the limit case, if you just take that toxin and put it on its own, where it's got no other molecules to interact with, it's not going to do anything at all, right? So what a molecule, any bioactive molecule, is capable of doing is context dependent. It's contingent upon its encounters with other molecules in specific types of contexts. That's the basic point being made here. Now, here is one of these assemblages, which is really just a cool example of something. This is the tobacco hornworm, Manduca sexta. And by the way, not some of these photos are mine, some of them are not. And I see that they're not credited because this talk was originally done for for an academic conference. I'm not making any money out of this, but if there are, if anyone sees their own photo in one of these slideshows, please get in touch with me. And I'm, of course, very, very happy to credit you. And I probably should have put the credits in the first instance. Apologies. So this caterpillar, the tobacco hornworm, it secretes a toxin which suppresses the plant defense response. So it's chewing on a plant, and in its saliva, it is secreting a toxin which suppresses that plant's defense response. The defense response of the plant when it gets chewed on is normally to locally increase the expression of its own defensive toxins. But the caterpillar doesn't want all those toxins in its food because they make you sick. So it is suppressing their production using its own countertoxin, right? Okay, we're already in a fairly complex dynamic here. It also is suppressing the production of these plant volatiles. I mean, this is actually its proximal mechanism of action, is it's suppressing the production of volatiles, so like pheromones, that the plant will release, which will tell the plant itself, other parts of itself, but also neighboring plants, to increase expression of defensive toxins. It's like, there are herbivores, alarm, 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 produce more defensive toxins. This is a way of that plants communicate relatively long distances more rapidly with themselves, with other parts of their own body, and of course with other neighboring plants of the same or sometimes even of different species. But the cool thing, well, another cool thing about this example is that the tobacco hornworm can't produce that suppressive toxin in its own saliva throughout its entire larval phase. Why is that? It's because adult moths detect host plants using the same plant volatiles. So adult moths are flying around and they want to know, where's a good host plant to lay my eggs on? They detect those host plants based on this production of defensive volatiles, of signaling molecules that the plant is producing to tell other plants to produce more defensive toxins. If those plant volatiles are not, if those signaling molecules are not in the air at some concentration, the adult moth can't find the plant and it doesn't know where to lay its eggs. So the caterpillars need to suppress that defense response, but they better not do it too completely. Otherwise, there'll be a cutoff in the generations. The adults won't be able to find any host plants. There won't be any more caterpillars. Uh-oh. But parasitoid wasps get in on the loop here, right? because parasitoid wasps also detect these plant volatiles and use them as a signal of where to find a juicy, juicy tobacco hornworm to take off to their burrow or their nest and to stab with paralyzing toxins through their own stinger, which is a modified ovipositor in the case of a wasp, inject it full of paralyzing but not killing toxins so they can lay eggs on the surface of that meat that will be quiescent, that will be quiet, but will remain fresh because it's still alive until the baby wasp larvae, which are like grubs themselves, hatch and then eat that caterpillar alive, right? The wasps need those volatiles to detect the caterpillar. The moths need the volatiles to detect the host plant. The caterpillar wants to suppress them but can't completely, otherwise the moths get lost, so the parasitoid wasps can get in now and exploit. Caterpillars also sequester the plant toxins, so they are resistant, in fact, to plant toxins. There's no complete stopping the plant from producing its toxins, so the caterpillar does have to be itself resistant, and it sequesters them in its own tissue, making it more toxic to any other animal that's trying to eat it. They do reduce the toxicity of the toxins that they sequester. So there's all kinds of crazy molecular modification going on in these situations, like attaching and detaching attaching things to molecules, detaching parts from them, shunting them around the body. This is all really, really fascinating stuff. And I mean, it's really ripe for a sort of analysis in terms of technicity, which again, future videos. But they also, again, they increase caterpillar unpalatability so they are a defensive toxin for the caterpillar to spiders and other predators. Now, this also gets more complicated because humans are in the loop now, because Manduca sexta is in fact a pest. What does it feed on? It feeds on tobacco and other related solanaceous plants. And amongst the defensive toxins this pest species interacts with is nicotine, right? So the toxins that the caterpillar is interacting with and controlling and that are associated with the release of this plant volatile are also, highly addictive stimulants that are at the base of a multi-billion dollar industry for humans, right? So this is a complex assemblage in which these molecular operators or machinic elements, the toxins and the volatiles and things, are right in the middle there. Just think that's all kind of cool. But I'm, a bit of a boffin. Another quote now from Deleuze and Guattari's A Thousand Plateaus. The distinction we would like to propose between machine and assemblage, a machine is like a set of cutting edges that insert themselves into the assemblage, undergoing deterritorialization, and draw variations and mutations out of it. For there are no mechanical effects. Effects are always machinic. In other words, depend on a machine that is plugged into an assemblage and has been freed through deterritorialization. Okay, so we've got the relational ontology thing in the second part of this quote again. Okay, this is not mechanical because mechanisms, the mechanical is a parts, extra parts kind of relationship, right? It's not what Simondon would call transductive relationship. It's a relationship of transmission, he would say, between the parts where they are not necessarily, although, see his thesis on concretization for nuancing here, not necessarily modified by their interaction, like reciprocally modified and shaped. In fact, what Simondon's concretization thesis shows is that this parts, extra parts kind of atomizing model is not even adequate to the technicity, the genesis of technical reality or of technical objects, right? But we're not getting into that. We're talking about this machinic element, which is like a set of cutting edges. So it's unzipping, it's opening up a pre-existent machinic assemblage, it's deterritorializing it, it's cutting connections between the parts and freeing them up to create new connections, right? For a kind of novel re-territorialization. So the machinic element here, the set of cutting edges, is something that is freeing up a latent capacity, or not latent, well, it is latent, but it's suppressed or it's canalized, we would say, in sort of biological systems language, a canalized capacity for variation because if we're good Darwinians like Deleuze and Guattari, we know that variation is the nature of things. Variation is primary for Darwin. The overproduction of variation comes before selection. This canalization, like the organism level of an organism, say, with the organizational level, which keeps things directed towards particular or somewhat directed towards particular ends and functions and all of that. That is a form of selection, is canalizing that inherent tendency to vary. And the machine is coming in and disrupting that. It's a differentiator, it's a disruptor. It comes in, cuts those connections, and releases that capacity to vary, which of course is part of the heterogenesis, the genesis of novelty, of new things, of more difference, difference from difference. So here, and this is, the notion of machinic pharmacoecology to me aligns with what I also call disruptive pharmacology, although I didn't come up with that term actually. So the toxin, or indeed the teacher here, as a machinic particle, this is another sort of link into my lecture series on the human relationship with drugs. So toxins mediate antagonistic encounters by entering the physioeconomy of target organisms and reorganizing it, right? So they're just like these machines, right? They enter in, they have cutting edges, they disrupt things, they don't necessarily reorganize, they disorganize, which can either open up the possibility of reorganization, or of course it can cause the whole system to spin out and the organism to die, right? Disrupting set points is what we're talking about here, and occasionally installing new ones. So again, cue endless examples about parasites that install novel behavioral phenotypes in their hosts, right? So they are not just disrupting, they are disrupting, but then installing something new. Think of like Sacculina, the barnacle parasite of crabs. Sacculina will attach itself to a male crab in the abdomen, and it will secrete a bunch of hormone toxins and other things, and it will essentially emasculate that male crab and cause it to start manifesting the female behavioral phenotype. So that male crab is going to go up to the beach and it's going to stand in the shallows, in the ripples of the wave, and it's going to do its little dance. And unfortunately, I'm not recording video of myself for this talk. Otherwise, you would see me doing the crab dance right now. I'm a very animated gesticulator, but you didn't need to see that this time. Anyway, it's doing its crab dance. It's releasing eggs, right? That's what the female crab dance is for here, releasing eggs into the waves so that they can be widely dispersed. But the eggs that the male emasculated crab, which is the host to Sacculina, is releasing are, of course, Sacculina's eggs and not its own eggs. Sacculina has installed a novel behavioral phenotype in that crab using toxins. Now, something that really cool that does happen here a lot in nature, and we kind of talked about this when we talked about basic chemical ecology at the beginning, is a symbiotic slippage across the spectrum from an antagonistic relationship to a mutualist relationship, with endosymbiosis as the kind of limit case of that, like one cell eating another cell, and that consumed cell finding an environment that it can live in on the inside of that other cell and eventually becoming an organelle of that cell and blah, 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 right? So this is constituting novel assemblages, including novel organisms. So again, it is contingent in all these cases on this disruptive effect of the machinic particle of the toxins. Which disrupt one set of norms, but potentially lead to the installation of, or the evolution of, the co-evolution of a novel set of norms. So I call that the transition from disruptive pharmacology to normative pharmacology. This is really a big thing when I talk about addiction and all of that sort of stuff, and the role of drugs in shaping society and other things in my other lectures. So we're looking here at the creation of novel physio-economies. Remember, economy is the management of the home, right? So I say physio-economy, I'm talking about all those set points that are keeping the organism, kind of in a non-equilibrium steady state. This is a homeostasis thing, right? Although again, homeostasis is not a good model for a fundamental understanding of what organisms are and what they do. Cybernetics will not help you here. Ashby is wrong. That's another talk. Actually, I'm going to talk about autopoiesis in just a second. So, um, compare this to the transition from exo to endo, from external to internal, from ecology to physiology. And of course, here there's a very, very rich opening onto a discussion of drugs and society and the way drugs shape society in a kind of evolutionary manner, um, sometimes by being disruptive. So, when caffeine arrives in Europe in the 16th and 17th centuries, and all of the caffeine producing plants, coffee beans, cacao beans, and tea leaves all arrive in England in like the same decade. And before that, there's no caffeine, right? So you can imagine, right? They have a fairly big effect on the culture. Uh, and there are many, many examples of this. Of course, you can think about the psychedelic 60s and blah, blah, blah. When drugs are discovered by a society that hasn't had an historical relationship with them and thus doesn't have containers, sociocultural containers for canalizing their effects, it can be very, very disruptive and it can have very big change in society. And maybe it becomes normative like caffeine did, or maybe that's what we're seeing with psychedelics these days. Again, all this stuff gone into in detail in those other lectures, which eventually I will record versions of. Okay, quote here from Guattari, maybe the last one. Yes, so we're moving now to the critique of autopoiesis, which I can never leave alone. Quote, but autopoiesis, which uniquely defines autonomous entities, unitary, individuated, and organizationally closed, I put that in, I think, closed to input-output relationships, lacks characteristics essential to living organisms, like the fact that they're born, die, and survive through genetic phylums. Okay, Guattari is recognizing the importance of the phylogeny here. Back to the quote. Autopoiesis deserves to be rethought in terms of evolutionary collective entities which maintain diverse types of relations to alterity rather than being implacably closed in on themselves. So I've done quite a lot of work on the ineliminable necessity for alterity in ontogenesis. My talk at the ASCP, the same conference I gave this one at in 2024, in 2025 was specifically on this. From a perspective of physical theory, actually. I recently gave a talk on Mike Levin's Platonic Space Symposium, which you can see here on YouTube, which also, and I think I'll put that in the show notes, as we say, but in the notes for this, I also make that argument there. And I'm definitely channeling Guattari amongst many others when I make those sorts of arguments. Autopoiesis is a non-starter as a model for organisms, and really even people who are in the lineage of autopoietic thought now themselves very much realize that, as we can see in the disentangling of enactivism from autopoiesis in the last, I guess it's the last couple of decades now, but I think it's really, coming to fruition theoretically at the moment. Autopoiesis, Varela and Maturana's theory, is very, very close. It's second order cybernetics, right? And it's very, very close to first order cybernetics in some of its assumptions. And the particular way that closure is handled there, this derives, I think, from Ashby's definition of a cybernetic system as, yes, thermodynamically open, but information tight. Information tight. So cue the Simondonian critique of information theory right here, right? Information tight. I mean, I know what Ashby means. I'm not going to get into it, but it's not a very good definition of an organism, certainly. And of course, cybernetics proposed to be, definitional for living organisms. In some sense, it does capture the organismic aspect, right? The organices. It's a form of organicism, but it, of course, absolutely elides the body without organs aspect of organisms here. And again, it's a mechanical conception. And it's a conception in which there are pre-constituted individuals. So it's like saying, if you find an individual, it must be autopoietic. Give me the individual and I will tell you that I'll describe it in terms of autopoiesis. So it's thinking Well, it's really not thinking individuation at all, but in a Simondonian sense, it's thinking individuation in terms of the individual rather than thinking the genesis of individuals via a process of individuation, right? It's starting with a given individual, and this is the real problem. It can't teach us about ontogenesis, heterogenesis, etc. And that's unfortunate because the whole goal for Varela and Maturana initially in proposing this was to provide a definition of living systems. And indeed, to replace what they saw as the neo-Darwinian conception, to replace that with a new fundamental theory for biology, but it fails in that regard. And neo-Darwinism also fails in that regard. And neo-Darwinism partly fails in that regard because it's insufficiently Darwinian. You can see many of my other talks for that. So against autopoiesis, little asterisk, specifically in its rhetorical or polemical deployment. Organisms aren't organizationally or operationally closed and merely thermodynamically open. Now, there's a lot of complex stuff to say about closure. Closure is something that organisms kind of do, that they have to achieve, but it's always relative. It's not good to start with your definition of an organism as closure. You have to talk about how organisms achieve it and how it's always porous. Organisms are always, this is the chemical ecology point, because they have to remain open and not just thermodynamically open, but informationally open. Um, they are always constitutively susceptible to other organisms coming along and putting operators in them, right? This is what happens in chemical ecology, and it's sometimes antagonistic, right? They certainly can't be operationally closed because another organism comes along and inserts operators which operate in, a way that is really analogous or homologous to the endogenous operators that that organism has that maintain its own physiology, its own, quote unquote set points, and again reorganize them in this machinic way. And as we've said, many, many lineages of organisms end up fusing in mutualist ways, and there's all these coevolutionary dynamics going on also for the antagonism. There's so much amazing stuff to talk about in coevolution of toxin and target, etc. These relations very rapidly become constitutive because of the lack of closure, the lack of the possibility of closure. Right? All evolution is coevolution, right? And yes, Darwin, he knew that, he understood that. So yeah, organisms maintain diverse types of relations to alterity. They're constitutively open to reorganization. This is machinic animism, right? Versus autopoietic autonomy. There's relative autonomy. I'm not against agency and telos and all those things. We need accounts of those in biology, but it's not, certainly not absolute, of course, right? And this animistic conception, which Deleuze and Guattari are sort of developing here, I've talked elsewhere, you can see my talk, Life is Technical, Not Mechanical, on the footnotes to Plato feed from last year's Biophilosophy conference, where I get into animism to some degree as a relational ontology. And for someone like Simondon, it's ontogenesis before ontology, actually, so it's ontologies, etc. And I'm saying here, and I said here that these issues aren't solved by the organizational account. I don't think they were taken sufficiently seriously by the organizational account, which is a research program in theoretical biology, early in the inception of that and early in its own break from autopoiesis. I think that they are getting along to that in a lot of ways. And also, there's Robert Rosen's research program and people like Hofmeyr who've extended that, but there's still work to be done here. I mean, I think everyone's kind of on the same trajectory now. Like there needs to be an understanding of these diverse relations of alterity. This is sort of a reference to something from Hofmeyr's extension of Robert Rosen's account, actually. So organisms are open to formal causation from other lineages, not just their own. And again, that's part of what we've been talking about here. But this talk's gone on way too long. This was a 20-minute talk at ASCP. I think it's been a great deal longer than that today, but hopefully interesting. Okay, questions. That is the last slide for a conference presentation. That is a Stevens banded snake. Isn't it beautiful? Questions, you can ask questions in the comments. I'll read the comments and I will hopefully respond. So if you made it this far, thank you very much for listening. And, I'll put out some more stuff in the new year.